Next, I'd like to introduce another young BME faculty, Fanny Chapelin. Fanny joined us about 2 years ago. She was trained in imaging and in UC San Diego, I believe. So again, I don't want to cut short her time. So Fanny, take it over.
Good morning, everyone. Thank you, Doctors Inadnit and Thank for organizing this great event. Today, I would like to introduce some of you to some magnetic resonance imaging platforms that we've developed in my years to quantify cell therapy distribution, persistence, and efficacy. As many of you are familiar, a lot of stem cells now are used for tissue regeneration, in particular in the clinic now. Some of the bone marrow, some of the bone and cartilage repair applications have moved on to the clinic. And you're also probably familiar with adoptive cell therapy, where scientists are trying to use a person's own immune system, engineer it to be smarter, to be able to then fight tumors. So when considering using all those live cells in patients, there are definitely some concerns. And this is also a concern of the FDA. The FDA actually recommends for scientists and clinicians to incorporate means to determine cell fate when using live cells into patients. And this is to better determine the survival, engraftment, distribution, possible differentiation of those cells, and tumorigenicity. To do this, there are multiple imaging techniques available to track cells in particular. If we do want to go clinically and also to be able to see deep into the tissue, we really just have two options, either PET imaging or magnetic resonance imaging. Unfortunately, PET requires ionizing radiation, and therefore our method of choice has been MRI over time to really prevent giving more radiation and more harm to the patient. In most MRI images, we do still need contrast agents to light up certain cells or tissues of interest. In particular, the common contrast agents out there are composed of metal ions that will provide either positive or negative contrast compared to regular tissue. In the clinic, though, there really is only those positive contrast agents that are available. And here you can see a brain tumor being lit up by a gadolinium kind of contrast. In the past decade or so, new agents have also come up that are composed of fluorine atoms. The advantage of fluorine is that we don't have any of those atoms in our body, so we can do really background-free imaging. And therefore, the signal that comes up is really proportional to the amount of contrast that you've put in the sample. And I'll show you examples of that very shortly. So when you're trying to see cell fate in vitro or in vivo, you have two labeling methods of choice. You can either incubate the contrast agent with some cells in the flask, and based on how much the cells can take up that contrast agent, you will have increased signal change. The other method is to inject the contrast agent intravenously, and we've seen over the years that this labels primarily two types of cells, either bone marrow stem cells or macrophages. And we'll see how we can use these properties to apply it for clinical purposes. So early on, I looked at cartilage repair as part of tissue regeneration, and we had a model of a rat femur head where we would shave off the cartilage and then replace it with a stem cell transplant. And by either labeling the stem cells directly or labeling the macrophages via IV injection, we were able over time to monitor if there was any stem cell loss, stem cell apoptosis, or possibly immune rejection of against those cells. And this is a little bit of the data. When we looked at the labeled stem cells over time here, we were able to see via the contrast agent. And here we have a map showing the intensity basically of the signal to enable quantification over time. We could monitor how much of the cells persisted in the implant over time. In a model of apoptotic transplants where the cells were not viable to begin with in the transplant, and by labeling the macrophages this time, we were able to see macrophage influx and therefore darkening of the transplant over time, whereas a healthy transplant remained bright. Finally, in a matched and mismatched model where we varied the gender of the recipients over time, we were able to show as well that MR enables delineation between a transplant that remains viable over time versus a transplant that is being rejected, where you have darkening again of the signal over time. We also used those types of contrast agents for cancer cell therapy imaging, but this time I actually focused more on fluorine compounds. We used a model of flank tumors in mice and injected CAR T cell therapy. CAR stands for chimeric antigen receptor, where the receptor is engineered to recognize specifically the tumor surface receptor. And I optimized a labeling method to incorporate those fluorine atoms into the CAR T cells and monitor their distribution over time. So after systemic infusion, to begin with, I harvested all the tissues from mice and ran NMRs, which are one-dimensional MRIs, and quantified how many cells went to which tissue. And we saw that CAR T cells specifically home to the tumors and remained over time compared to a mock therapy. And that was, we had similar signal in the spleen over time, so we could see specific homing of this therapy. Unfortunately, we cannot harvest tissue and we don't want to biopsy patients all over when we think of the clinic. So one of the strategies was to improve the perfluorocarbon design so that we would have better signal in vivo. And we did manage to do that by incorporating cell-penetrating peptides. And here you see locally injected T cells with our new contrast agent versus the existing fluorine contrast. Finally, we used a very neat property of the perfluorocarbons, which is that they can, in a sense, sense the local oximetry in the tissue. And so this time we labeled the cancer cells before infusion into the mouse and measured the oximetry based on whether they received the CAR T cell therapy or the mock therapy. And as you can see, we were able to see that as the cells reach the tumor, there is increase in oxygenation in the tumor, indicating non-invasively that the tumor is responding to the treatment. Whereas here, both untreated and mock therapy receiving mice did not have any change in oximetry. And this is a very interesting avenue for at least preclinical research, but when we consider clinical research, we do want something that is applicable in the clinic. Unfortunately, there are currently still very few fluorine MRIs available in the clinic, and this is the reason why, when joining UK, I've also developed a collaboration with Drs. Gedali and Marty. Dr. Gedali will speak just after me, but to be able to track T cells in the clinic, so not with fluorine compounds, but back to more proton type contrast agents. And I cannot go into much detail, but we are investigating a new agent and compared it to what is the gold standard in a sense in cell tracking in our field, ferumoxidil, and we saw similar types of signal enhancement. And we were able to label successfully our T cells with this agent, as depicted by the little blue dots here in the cells. And when we quantified the signal of the labeled cells via MRI, we did see a good shift in the signal in as few as 500,000 cells compared to unlabeled cells. And so we are really excited and looking forward to tracking those cells now in vivo. And this is what is ongoing at the moment in the lab. With this, I would like to thank you for listening, thank our collaborators, specifically in the School of Medicine, my students, and our sources of funding. Thank you very much.
Dr. Chapelin, thank you for a very interesting presentation, and it's great for us clinical surgeons to see really bench work being translated into clinical activity. So congratulations.